Our next speaker is Sheena.
Great, well, thank you. Had a great meeting so far, so thank you all for stimulating all the discussion. I don't want to thank whoever it was that sent a little gremlin into my room and completely killed my laptop while we were out for dinner. So I spent most of the night trying to get this talk on the internet and then onto a memory stick, and here I am. So I actually don't know what's going to happen. So if there's any IT gurus in the audience, I can't open anything Microsoft. So how do I get rid of this thing at the top? I don't. Okay, right. Okay, so I'm going to, is that okay for everyone? Yeah, I'm going to talk a little bit about going back to sort of the basic principles of why and how proteins fold and misfold. And I think it was probably worth starting. I was quite pleased when I was thinking about how to sort of set the scene for this discussion. I haven't got a huge amount of data. I wanted to try and make you all think. In that actually it's 50 years since Amundsen won his Nobel Prize for the principle of protein folding. And I think many of us in this audience are here because of that work. And of course what we learnt from Amundsen is that the unfolded protein not only defines the structure of the native state, but it defines the search or the process by which the native state is sought and found from the billions of alternative possibilities. And it's a theme I'm going to come back in a little bit when we come onto protein aggregation. So then thinking about, well, what started the protein folding field off? It was, of course, started by Sanger being able to sequence the protein sequences using Edmond degradation, and the start of crystallography, where we started to see for the first time where the polypeptide chain was going, where the ultimate structure was. And David Eisenberg reminded me over dinner last night that in 1965 there was only 2 structures solved by crystallography, so it shows how rapidly the field had moved. And of course, what the protein folding field, I think, gave us was the concepts that proteins fold on these energy landscapes that you've all seen. But I think what it really taught us, it gave us the tools and the techniques to think about how to define non-native states of proteins, everything that crystallography couldn't give us. Partially folded states, how to define the conformational ensemble of the unfolded state. We had NMR methods, single molecule FRET methods, hydrogen exchange NMR, hydrogen exchange mass spec, and obviously computational methods that have allowed us to put all of that plethora of data together to define how a protein sequence defines the search to the native state. Now, what we know in protein folding is we can abuse our proteins a lot. We can make mutants, we can change pH, we can change conditions, and they will still circumnavigate their landscape to find the native state. So, of course, we don't just have a few sequences. Now we have over 200 million sequences from organisms I've never heard of, and the PDB close to 2,000 structures. And, of course, 200,000 structures. And, of course, what that has given us is another revolution that, of course, we'll all be using in our labs is AlphaFold, which is using the information from sequences and structures to predict protein-protein interactions. So with that sort of way of thinking about things, when you come to think about protein aggregation, it's just the same thing, but a lot more complex. And it's complex because any species on this reaction coordinate can go off and do gymnastics and make friends with other molecules, whether they're proteins or RNAs or membranes or whatever, and form what I've put on this side of this diagram, sort of amorphous or disordered aggregates. And I put on the other side, just because it's the easiest way to draw the picture, aggregation via oligomeric states to form protofilaments, fragments, and fibrils. And we know that all of these species have different effects on cells, and we know they're all populated together. But what we don't know is which of these species form amyloid, which of these aggregates form amorphous aggregates, which are toxic and which are not, which are on pathway and which are off pathway. And so we need to kind of use what we've learned in protein folding, I think, to map all the species that a polypeptide chain can make, not just homotypic, but heterotypic, if you like, to understand the relationship between sequence and the aggregates formed. And the reason it's really hard to do this is we don't have the chemical probes or tools to do a structure-activity relationship. What you'd want to do is add a ligand to take this guy out of the reaction and see what happens in a cell. Well, we can't do that unless we understand the structure and dynamics of these species more. And so which species do we target to treat disease? And in vitro versus in vivo, of course, it's yet more complex. And so what we know then for neurodegeneration, of course, we're mostly talking about aggregation from intrinsically disordered polypeptide chains. And rather than folding to a unique native fold, these disordered chains can aggregate to many different ultimate structures. So it's not one sequence, one structure, it's one sequence, many structures. And the important point is that whilst protein folding is under mostly thermodynamic control, you can abuse the sequence and it will find the native state, aggregation to amyloid-like fibrils is under kinetic control. And that means it's incredibly sensitive to the sequence, to the solution conditions, and to any other species a polypeptide chain may find on the reaction coordinate. Chaperones, RNA, membranes, metal ions. We've heard about all of these things. And so, of course, in vitro, that gives us an array of amyloid folds for the same precursor sequence. This is some of the 31 different synuclein amyloid fibrils solved by cryo-EM. And so what we really want to understand is how the kinetics of aggregation defines the species that are formed and the ultimate products. And so that's what we've been trying to do in the lab, and we haven't done it. And I don't have the answer to all of this, but I'll tell you what we've been doing. So what we've been doing is what I call top and tailing the question. So we want to link aggregation kinetics, which species are formed, how fast are they formed, and how are they interconverting, to the structure of the fibrils formed. Here is 3 polymorphs of amylin solved in Leeds in the Ashby Biostructure Lab with my colleague Neil Ranson. So what we really want to see is how the rate of fibril formation changes the species formed and the final fibril product. So we want to look at how aggregation is initiated. Can assembly be arrested by targeting these early interactions and push species away from toxic intermediates, or to define fibril products that may affect cells differently? And I think that's the kind of thing that the NSF might like to think about funding. So for synuclein, it's 140 residues, and others, but using methods to try and define what the IDP looks like. You all know the sequence of IDP, the central NAC region, and all the mutations called in familial disease are not within this region that's necessary and sufficient for aggregation, except this E83Q that Hilal and colleagues identified recently. David Eisenberg talked about this region we call P2 as the pre-NAC region. So what do these familial mutations do? Well, clearly, and this is a really nice paper, they look at the rate of fibril amplification on membranes and off membranes. And all you need to see from this is there's no correlation with these mutations and the rate of aggregation in the different environments. And these mutations, of course, are not within the amyloid core of the protein. They're in these so-called flanking regions. So what are they doing? So what we're very interested in doing is having a look at these flanking regions that form these hairy end or furry coats on amyloid fibrils, about 80 residues in synuclein you don't see in the EM structure. And we want to look at how these flanking regions affect assembly. How do they affect the assembly mechanism, and how do they affect the structure of the fibril formed? So today's story, just to emphasize one way we've been doing that, we started looking at synuclein. We did some dynamic force spectroscopy to map at a single molecule level where the first protein-protein interactions of synuclein were. And we mapped them to residues 40 to 60. We had a look at residues 40 to 60 using Zaggregator for amyloid propensity and CAMSOL for solubility, both written by Michele Vendruscolo. And we identified these regions. So, of course, the NAC has a high aggregation propensity and low solubility. So does the pre-NAC region that David's already told you about, which we call P2. But this little region, P1, jumped out to us. It has a high amyloid propensity and a low solubility, but it isn't picked up by David's ZipperDB program. So we wanted to have a look at this bit of the flanking region in aggregation, and it has this rather boring sequence, GVLY, VGS. So here are some aggregation kinetics at lysosomal and neutral pH of wild-type synuclein. A synuclein lacking just this 7 residue sequence that's N-terminal of NAC. And you can see this has a dramatic effect. It slows nucleation at acidic pH and ablates fibril formation over many days at neutral pH. We don't see any fibrils by a whole range of techniques. Rather than deleting P1, if we replace it with an SG-SG-SG linker sequence, we have the same effect as deleting it. And so this tells us that this little sequence is necessary for rapid, if you like, nucleation of aggregation. And of course here it also emphasizes how the solution conditions are incredibly important for defining aggregation. It's a kinetic control. More recently we've run an alanine scan through this P1 region at neutral pH, and your eagle eye will spot easily that there's just 2 residues. If we make Y39 to an alanine, that's quite a large substitution, but serine to alanine had a dramatic effect on nucleation. And we know it's nucleation because all of these concede fibril elongation. So this tells us that a single residue is required for nucleation, at least on this biochemical timescale. Now, never say never for amyloid formation. You can see a small change here. Is this a biophysical phenomenon? It isn't. We can put these variants into C. elegans with the Wkshp flank and show that while we see puncture in the wild-type worms, these small substitutions stop puncture formation in worms and they start wiggling again. And so I think this shows you the critical balance between the rate of nucleation of an amyloid fibril and the ability of an organism to deal with it, so that small changes in the rate of nucleation can have a dramatic effect on phenotype. We've also gone further to look at other mutations in the protein, actually inspired by the difference we've seen alpha-synuclein, which is amyloid forming, and gamma, which is more aggregation resilient. I just want to show you this one, L to M, at position 38. So here's alpha-synuclein with leucine 38. If we put methionine in, a dramatic decrease in the rate of nucleation, and a simple leucine to isoleucine can speed up aggregation. And so what I wanted you to take home from this is that these very precise amino acid substitutions can have dramatic effects on the rate of aggregation and the organismal consequences for that by tuning the rate of nucleation. So finally, does any of this matter? Why has synuclein got this sequence that makes it form protein aggregates? Well, it turns out that this sequence is vital. I'm showing you the data. It's vital for membrane remodeling, but not for membrane binding. And it's also important for chaperone binding and sumo binding. And so biology has given a sequence that it uses for functional reasons, but the downside of that is aggregation is enhanced. So finally, can you target aggregation by looking at these small flanking regions? And this isn't our work, this is my last one row hit. This is some very nice work from Wolfgang Hoyer and Alex Buell, who made this beta-rapin nanobody that binds to the P1 region and shows that it prevents from aggregation in vitro in neurons and in flies. And this P1 region also contains a sumo binding site, and binding of sumo to this region also inhibits nuclear aggregation in vitro in cells and in flies. And so my take-home message really is that the energy landscape for amyloid formation is complex and it's under kinetic control. I think we need this integrated toolbox of methods like we learnt for protein folding to look at sequence mechanisms and how the rate of aggregation alters the products and the effect on cells. That these flanking regions that we don't see in electron micrographs can be very important both in controlling the rate of aggregation and the fibril products. And these large region fibrils remain dynamically disordered and could be interesting targets for disease recognition and modulation. Thank you.
while our next speaker is setting up, we have time for one question here. Richard?
Really nice story. So how do you think P1 mediates nucleation? Through interactions with itself or with other regions of alpha-synuclein?
Yeah, we did a whole load of NMR experiments, and so P1 interacts with other molecules in the P1 region, and if you add a P1 peptide in trans, you can actually recover from the loss of the P1 region. So I think it's mediating those very early protein-protein interactions that we saw in the dynamic force. But the take-home here is that those small changes in the rate of nucleation can have dramatic effects, at least in C. elegans.
All right, Giovanni, while you're setting up Hilal, you can ask one more question quickly.
Thanks, Sheena, for a beautiful talk. So these regions are, first, some of these residues are visible in the MSA structure, right? Because it starts at residue 39. But these residues are all in the membrane binding region. Have you looked at the effect of these mutation in membrane-dependent aggregation reactions, right?
Yes, we have. So these regions, if you delete these regions or mutate these regions, you change the binding affinity to membranes by about tenfold, but they still bind membranes. They don't do membrane remodeling, but the membranes can still cause the lipid accelerated rate in aggregation. So they still do have a kick in the rate of aggregation of the membrane surface. But the mechanism they do that is very different. The structure of the membrane bound state is different.
Awesome. Thank you so much, Sheena.